Okay, let's start with lecture 2, okay? Lecture 2. Let me know if I need to slow down a little bit, okay? We do have a lot to cover, so you know, if you miss something, you let me know. I will post this whole thing online as well afterwards, not just the recording, but the notes as well. But like I said, like 10 times already, please try to copy along. So in lecture 2, which is macromolecules, we talk about, you know, basic elements, right? Basic elements in the body, right? So these include things like carbon, hydrogen, oxygen, nitrogen, and phosphorus, right? So these are the common elements in the body, okay? They are the ones that makes up a majority of the molecules within the body, okay? And of course, the important thing to know is when we discuss macromolecules, you have to know what atoms make up what macromolecules. And we're gonna do a summary of that in a bit. Okay. Then we talk about trace elements, right? Trace elements. These are trace means is only found in small quantity, right? Only found in small quantity, right? Small amount. And you know, we talk about things like iron, which you need for hemoglobin. There's iodine. There's magnesium, right? So some examples of trace element. Make a list, right? So you will know that they are trace elements, and you only need a little bit of them, okay? Not too much. Other things we talked about would be things like hydrophilic, which is water loving. So water loving, as well as hydrophobic, okay, which is water hating, so to speak, okay, water repelling. And the key thing to sort of know here is that hydrophobic and hydrophilic things, they don't mix, okay, they don't mix together. Okay, and the reason we have to know that is because knowing hydrophobic and hydrophilic substances will not interact well, we can predict what's going to cross the cell membrane. Okay, so the cell membrane has the phospholipid bilayer, which has the hydrophobic core in the middle, and so if you are a hydrophilic molecule, a charged molecule, you won't be able to get through that easily. Okay, so that's that. We talked a little bit about pH. Okay, so you know, if it's acidic, something that's acidic, then it has high H plus concentration. Square bracket means concentration, which means is going to be less than 7 in pH. Okay, and then neutral is equals to 7. The pH is equal to 7, and the opposite is basic, okay, which is low H plus. Okay, and that is going to be greater than 7, Okay, so that is pH. And you know, in terms of why we have to know about pH, well, pH affects the shape of proteins, right? Shape of proteins. It affects rate of diffusion. Okay, so things that we talked about in later lessons. Okay. Then the big thing about this unit is monomer monomer and polymer. Okay, so we know based on our discussion that from monomer to polymer, we have to do dehydration synthesis. Okay, and based on what we learn in metabolism, we know that linking monomers to form polymers, that is going to require energy, right? That is going to require energy, and so this is part of anabolism. Part of anabolism require ATP. Okay, whereas the opposite, which is breaking down polymers into monomers, that is going to be hydrolysis. And that is part of catabolism. Okay, and it will generate energy. It will generate ATP. Okay, just stop me anytime if you have questions.
Yes. What are isomers?
Oh, okay. So isomers, they are basically two molecules. Okay, so isomer is not really related to this, right? Okay, it's kind of like the earlier stuff. So two molecules with same chemical formula, but different structure. Okay, but different structure. So for example, I think I might have done this in before. So let's say this molecule, right? This molecule. Okay, this one, if you count the carbon, is C4 and then H10. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. But I can also draw it in another way. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, like this, right? So they are both C4, H10, but the chemical structure is different. So that's what an isomer is. Okay?
Okay, thank you.
No problem. All right, moving on. In terms of, again, right, back to the monomer to polymer, right? Monomer to polymer.
I have a question.
Yep, sure.
Dextrorotatory and lever rotatory are also isomers.
Yes, so what you're talking about is a different type of isomers. I don't think I talked about that in my lecture, but there are different types of isomers. Okay, like this one is called structural isomers because they are, structurally connected differently. The one you're talking about is like a mirror image. Okay, and since you brought it up, I'll just quickly talk about it. Okay, so let's say I have a carbon, and then it's going to be connected to four things. Okay, so this means it's sticking out a page. This means it's into the page. So I can have four different things here. Okay, and what happens is I can have another molecule that is going to be exactly mirror image of that. Okay, so they are not the same molecule. They're kind of like the left hand and the right hand, and that's kind of what those terms you mentioned mean. Okay, so we have, oops, sorry, that should be a different color. Okay, so we have a right hand configuration and we have like a left hand configuration, and then they are mirror image. Okay, so it's also an example of an isomers. We call these enantiomers. Okay, so monomers to polymer. Okay, we did this question before, but let me just remind you. Let's say I have I'm making this up as we go. H, okay, and then there's an OH here. Okay, let's just like that. And then I'm gonna put another molecule here, OH, C, H, H, C, H, OH, H like this. And then I ask you how you can combine them to form a link. Okay, so you should know that you are going to be taking away OH from one, you're going to be taking away H from another, and that's how you remove the water molecule. Okay, so afterwards you're going to have C, the exact same thing, but now you're going to connect directly to the O. Okay, like so. Okay, so I think there was you can check back the week 2 tutorial. There was kind of like a poll question that we did. That's like this. Okay, so this here, this is the new bond. Okay, this is the new bond. Okay, just let me do another example with you. You remember amino acid, right? Amino acid. So in amino acid, we have C, H, C double bond O, OH. This is the R group. And then this is the amino group. Okay, so over here, amino group. Okay, over here, this is the acid group or the carboxyl group. Okay, so what happens is if you take one amino acid and you put it next to another amino acid, okay, this is R2, this is R1, because the R is what distinguishes them, right? Okay, so what's going to happen is you will take the OH away from this one and you take the H away from that, pulling out H2O, and then you're going to form a new bond. Okay, this is the new bond that connects them. Okay, the key thing to know here is this: doesn't matter what molecule I give you. Okay, if I ask you to link them together and I ask you what the results are, you will always be removing an OH from one and an H from the other, and then you connect them together. Okay, any question about this? All right. So let's do an overview of the macromolecules. Maybe we can do a chart or something. Okay, we'll do a chart. Okay. So this is macromolecule, and then we will do elements here, and then we'll do type here.
A question, sir. Can you go a little bit like upwards, like the last question?
This one, yeah.
A little bit up. Okay,
this one?
so it's is it? Okay, so the second one, which is this one?
This
is
one?
Yeah, it's like a. What kind of sorry, I missed it. That's why, like, what kind of compounds like this? Those two are like.
Oh, I was just saying, right? Like, you don't even have to know what they are. The point is, you know, this C is basically this C, and then this C is basically this C, right? The important thing here to know is if the question asks you to link something together, to get the answer, you would take the OH off one of them, and then you would take the H from the other, and then you just, you know, create a link between them.
So okay, and between the amino acids, it's like a peptide. It's like a bond, right?
Yes, between the amino acid, this new bond is the peptide bond. Okay.
Thank you. Thank you.
No problem. Okay, so macromolecule. We have carbohydrates, for example. Carbohydrates. Okay. So the elements in carbohydrates include carbon, hydrogen, and oxygen. Okay. So just very generically speaking, the types we have monosaccharide. Okay, so monosaccharides would be like glucose, fructose, And there's one more. It's galactose. Okay. Then, of course, you can link these together in various combinations to create the disaccharide. Okay. So disaccharides, we have maltose, sucrose, and lactose. Okay. And then one level higher up in this would be polysaccharide. Okay. So what are some polysaccharides? Anybody? You can just, speak up if you want.
Cellulose,
Yep. There's
glycogen,
cellulose. Very good.
starch.
Yeah. Very
Starch.
good. Good. So cellulose, glycogen, and starch. Thank you. Okay. So of course they all have different functions. We'll summarize the function in another chart, perhaps. But that's basically it, right? For carbohydrates. Moving on, we have lipids. I think that's the order I did it in for the lecture. Lipids is also CH and O. And I remember there was a question that asked you, out of the 4 macromolecules, which one is not composed of repeating units of monomers, right? And the answer is lipids. Okay, so carbohydrates, you put many glucose together, for example. Proteins, you put amino acids together. Nucleic acid, you put nucleotides together. But for lipids, we are putting components together. So they're not really like a train, okay, of repeating units. Okay, they are just several parts that are coming together. And the types that we talked about include triglyceride. Another name for triglyceride is fat. Okay. We also talk about phospholipids. Okay, and again, we will summarize the function in a later chart. Okay, otherwise the chart's going to be too big. I'm just going to make this longer. We also talk about steroids, as
sorry,
well as. Sorry.
I have a question. Sorry.
Yep. Sure.
So if there are questions saying what are the elements in lipids, so say that you said like CHO, right?
Yeah.
But in phospholipids and steroid, like you have like phosphate groups and nitrogen groups. So so the question.
Yeah, that's a good question. Okay. So I can put here, trace of phosphate, right? Trace of phosphate. I'm glad you mentioned that because I think there's a question like that. Okay. And so for lipids, it's important to remember we do have phosphate in the phospholipids. Okay. Because the phospholipid has the glycerol, the 2 fatty acids, as well as a phosphate group. Okay, in the head. Okay. So please remember that. Okay. And then there is wax as well. Okay, so those are the different types of lipids. Okay. Next, we have proteins. Okay, proteins, we have C, H, O, and N, and then I'm gonna put star as well. There is also a trace of. Anybody know? Trace of what? What's the extra element that I'm looking for?
Like sulfur.
Sulfur. That's right. trace of sulfur. So in one of the amino acids, one of the 20 amino acids, the cysteine, okay, that's the one that has the sulfur in it, okay, and that's going to be important to know for, you know, one of the lectures in the second half of the course. But you know, for now, just kind of make a mental note of that. And you know, we have different types of proteins. We have like proteins that are enzymes. We have proteins that are in the membrane, okay, either fully embedded or just attached on the side. And we have hormones as well, hormones. And you know, in terms of lipids here, the steroids, there are cholesterol, and hormones. Right. Some of the hormones are made from cholesterol. Okay. And lastly, we have the nucleic acid. Nucleic acids, which has everything in it. Okay, CHO, N, and P. And there are only two types. There are only two types of nucleic acid: the DNA and RNA. So that is that. Are we okay so far?
I have a question, sir.
Sure.
Sorry. So for the proteins, can we talk about the denaturing of the structure as well?
Yeah.
Remember we
Sure.
having questions on the physics and yeah.
Yes, we will. We'll get to it for sure. Okay. Thank you. Okay. So now that we have a overview of all the molecules, let's go and do individual charts for these things. Okay, so let's do carbohydrates. So let me think about what kind of chart to do here. Okay, so someone asked, we will have a break today. Yeah, we're gonna have a break at like the usual time, like 1 hour or something, so you can rest your writing muscles. So this is type, and then we will have I guess examples and function. Okay. So types. So we talk about monosaccharides, right? Monosaccharides, which is a single sugar. An example of that would be glucose. Okay. So there are other examples as well that I didn't put in the earlier chart. But in the context of what we're talking about, I think it's good to include, for example, ribose and deoxyribose. Okay, so anything that ends with OSE, it's likely a sugar of some sort. Okay, so glucose, fructose, galactose, ribose, deoxyribose. And the function of these things would be energy source, right? Energy source, right? So in cellular respiration, I'm trying to link the units together for you, right? Cellular respiration, and that is basically what the glucose is for. What are these for? Anybody remember what these are for? Like if I have to state a function for these sugar, what would you say the function is? Anybody remember, recognize ribose and deoxyribose? Can either type the answer.
For like DNA structure.
Yeah, exactly, right? It's for building up the DNA, right? As well as the ATP as well, right? Okay.
Yeah.
So we will say these are building blocks. Okay, building blocks for DNA, RNA, ATP, that kind of stuff. Okay, so that's how they are related. And sorry, just gonna make the chart bigger. What else do we have? Well, we have the disaccharides, but then the disaccharides is pretty much the same thing. It's like energy source. Okay, so I'm not gonna write another one. I'm going to go to the polysaccharides though. Okay, polysaccharides. Okay. So someone mentioned starch. Okay, so starch is for energy storage, mostly in plants. Okay, humans don't have it, of course. There is glycogen, which is energy storage in muscles and liver. Also, someone mentioned cellulose. Cellulose is for structural support. Structural support, like in plants, for example. And there's actually one more polysaccharides that we talked about. Chitin, that's right. Okay, chitin is for the exoskeleton of insects and shellfish, that kind of stuff. So exoskeleton. Okay, so that is basically it for that. There is also another function for polysaccharides, and again, I'm trying to connect the units together. We don't really have a specific name for these stuff. We just generally call them glycoproteins. Okay, so glycoproteins are proteins in the membrane with some sugar stuck on it. Okay, and the function of glycoprotein. Anybody remember function of glycoprotein? This was
Like
lecture 4.
gives the identity.
Yeah. So there
It's
is. Yeah,
a
go ahead.
protein with carbohydrate attached on it.
Yes, and the function of that would be.
Like giving identity to cells and like
Yeah, exactly, right? So cell recognition. Cell recognition. Okay, so someone also said like cell signaling earlier. That's not really glycoprotein. It's a function of membrane protein though, but specifically for glycoprotein, in the context of what we've talked about, this is for cell recognition. So if you wanted, if a white cell comes and want to distinguish what belongs and what does not belong to the body, then it would, use the glycoprotein as one of the surface marker to identify that. Okay, so that is carbohydrates. I mean, let me see if there's anything else I want to add here. In terms of structure, right? These are for lack of better word, these are like little hexagons, right? Hexagons, right? And pentagons, right? They are actually called rings, okay? But if you look at the pictures, they're hexagons and pentagons, right? But for polysaccharides, depending which one you're talking about, right? They are either, long linear chains or, with some branching. And then when we talk about things like glycogen, they would be extensively branched, okay? So, linear, okay? Some branch, and then this is extensively branched. Okay. I hope this is helping. I'm just kind of thinking about the stuff as we go. I hope I don't miss anything. Next, we should do lipids, maybe? Lipids. Okay, I gotta go faster. We have lots to do. For lipids, so let's do another chart. We can do types of lipid, types. We can do components. We can do examples and functions. Okay. Oops. So types, what do we have for types? Fat, right? Triglyceride. triglyceride fat. And to make fat, you need the following components. You need 1 glycerol, right? Glycerol plus 3 fatty acids. 3 fatty acids. And the fatty acids could be saturated or unsaturated. We'll review those in a bit. And an example of that would be, well, like a butter, right? Butter. Is butter saturated or unsaturated? Actually, you know what? I'll just answer that myself. It's saturated. And we have things like, you know, olive oil, right? I guess that would be unsaturated. Unsaturated. Okay. So what are the general function of fats? They are actually pretty good for energy storage. Energy storage. Okay, so if you are starving, if you haven't been eating for, I don't know, 2-3 days or something, you will start to burn off your glycogen reserve. Then the next thing that your body will turn to is going to be fat. Okay, so you can chop up the fatty acids and you can feed them into Krebs cycle and generate ATP and NADH that way. Energy storage and for insulation, right? Insulation. So keep you warm, basically. So that is a fat. That is a triglyceride. There is the phospholipid, phospholipids. And the components for that is also 1 glycerol plus this time 2 fatty acids. 2 fatty acids. Okay. Usually is 1 saturated and 1 unsaturated. But they could both be saturated as well. And very importantly, it's going to have an additional phosphate group. Phosphate head, we call them. Okay. The fatty acids are the tail, right? Fatty acid tails. Okay. So the tails are the hydrophobic part, right? Hydrophobic, and the head is the hydrophilic part. Hydrophilic. Okay. An example would be the phospholipid bilayer. And the function is form the cell membrane. Form the, you know what, I'm just going to say cell membrane. Cell membrane. Okay. So I'll give you a minute to catch up in case I just realized I'm writing a little bit too fast, probably. I'll give you a moment to catch up. Okay. Meanwhile, if you have a question, ask away.
So I'm so sorry, this is not from this chapter, but when you said cell membrane, I remembered. When in is it endocytosis?
Sure.
You know, the cell membrane forms a kind of a vesicle.
Yeah.
I remember there was a question in the quiz, like, what happens to the cell membrane at the end? Is it inside the vesicle, like something like that?
Yes.
So can you kind of clarify that? It's like at the end, it's inside the vesicle, right? Is it?
The membrane is going to be. I don't remember. I don't remember exactly which question you're talking about, but okay. So
like I think it says at the end of the reaction or something, what happens to cell membrane, where it is, like inside, outside of the vesicle, something like that.
So let me try to explain this. So it's phospholipid by layers, right? So let's say this is the outer layer and this is the inner layer, okay? Okay. And this is the outside of the cell, and this is the inside. Okay, so let's say there is some particle here that you want to take in. All right, so what's gonna happen is this. Okay, what's gonna happen is this. Like that, and then like this. Okay, and then you're gonna have the particle here, and then oops. So this is 1, this is 2, and then finally, when it's all said and done, then it's gonna be like this. Okay, so like this part and this part is going to to connect and seal together, and then your vesicle is going to come in here. So the outside, which is the blue here, right? The outside of the phospholipid will now be the inside of the vesicle. Okay?
Yeah, that's the question. Yeah, thank
Okay,
you.
no problem. Okay, so we're talking about lipids. I think I'm just gonna erase this part because I want to continue my chart. But if you need to watch that again, just look at the recording. Where are we? Phospholipids. What else, guys? Phospholipids. We also have steroids, right? Steroids. And the components of steroids is basically, you know, 4 fused carbon rings, okay? And then you add a bunch of other stuff to it, okay? Other functional groups to it, but the core structure would be the 4 fused carbon rings. And examples of steroids would be hormones, okay? Hormones such as testosterone, estrogen, right? These kind of stuff. And the function would be, we'll say reproduction in this case. Okay, reproduction. And you know, signaling. Okay. Lastly, wax. Just very quickly, wax. This one is going to have a long fatty acids plus some carbon rings, and plus I think in the lecture notes I mentioned something about alcohol, right? Alcohol is not necessarily what you think, okay? Anything with OH in it is an alcohol, okay? So anything with OH. Okay, you'll learn more about this in organic chemistry next semester with your chemistry teacher. But anything with the functional group OH is going to be classified as an alcohol. An example of that would be, you know, the waxy stuff on top of plants, right? We call that cuticle, so surface of plant, and the function is water resistant, water repelling.
Question.
Yeah.
So in wax, so at one time all these 3 components are there. Is it?
Yeah. So it's going to be linked to either alcohols or the rings. Okay. So I guess I should say or here, or the rings. Okay. It could be both. It doesn't have to be both.
Oh, thank you.
And that's it for lecture 2. Okay, that's it for lecture 2. So let's do some polls. You know, maybe we won't be able to have the break. Sorry, Emily, you asked about that earlier because this is taking longer than expected. But you know, if you need a break, do take a break. If anyone need a break, take a break. And you can catch whatever you missed later on with the recording. Okay, so let's try some poll questions. Okay, let's try to keep it under a minute, guys, so we can, you know, move ahead. Okay, there's the question. Okay, a couple more seconds. Okay, so this question is about basically saturated fat versus unsaturated, right? So how do I even write on this? Give me a second. So saturated, right? Saturated versus unsaturated. And this is going to have a kink and a bend in it. So it's talking about the saturated fat. So a couple of things about saturated fats. Sorry, it's going to be unsaturated fat. I mean, okay. So a couple of things about unsaturated fat. Number one, it's going to have a double bond in the structure. That's what's creating the kink. It's going to be liquid at room temperature because of the kink. The kink will cause the molecules to not pack together closely. And so it's going to be a liquid at room temperature. And most of these fats are plant derived fats. Okay, so what's the answer? Well, as I just mentioned, it is saturated. It's unsaturated fat. So this is wrong. This molecule must be a phospholipid. That's not true. I just mentioned earlier, right, when we were doing the chart, that the phospholipid tail could be both saturated or one saturated, one unsaturated, right? So just because it has a kink in it, we don't definitely know it is going to be a phospholipid. And in fact, it might be just a regular triglyceride, okay? This fat would be solid at room temperature. Not true, because we satisfy that it's unsaturated, so it's liquid at room temperature. And the kink is created by the double bond. So the answer is D, okay? Next one. Okay, let's try this one. I'm going to relaunch the poll. Okay. There we go. Oh, sorry. I launched the wrong poll. Give me a second. This one is multiple answer. I have to launch the multiple answer poll. Okay. There we go. Okay, try again, please. Sorry. I meant to say phosphorus, not phosphate. Sorry, I just made a mistake there. Phosphorus, okay, not phosphate. Okay, anyone else? All right, I'm gonna stop the poll there. So if I'm talking about phosphorus, okay, phosphorus is P, right? Okay, then the answer is all of them except for A. Okay, they all have phosphate in it, phosphorus in it. If I'm talking about phosphate, okay, if I'm talking about phosphate, then the answer is just going to be oh, sorry. I'm confusing myself. Whether I'm talking about phosphate or phosphorus, the answer is just C, D, and E. Okay, a protein doesn't have any phosphorus or phosphate in it, and starch doesn't have any. Okay, so it's just C, D, and E. And the difference is phosphorus is P, and phosphate is PO4. Okay, so the phosphorus exists in the form of phosphate for both C, D, and E. Okay, so in DNA and RNA, the nucleotide, they all have a phosphate group in it, right? So again, the correct answer is C, D, and E. Okay. Very good. Next one. Okay, this one should be fairly straightforward. Let me launch the poll again. Go ahead.
So like that's two A's, right? Can I just say second one is B?
Oh, but then we're waiting for everyone else, right? So we gonna have to.
How can we choose?
Okay. So this one is talk about the base pairing rule, right? And it's between DNA and DNA. So you have to know A pairs with T and C pairs with G. And I just remember someone earlier in the chat told me to review purine and pyrimidine. Okay, so purine, they are the adenine, and guanine. Right? I said the acronym for that is a pure as gold, right? That's how you can remember that. And then for the pyrimidine, it is going to be pyrimidine. It is going to be the thymine, and cytosine. Okay, and of course that includes uracil, as well. Okay, so in terms of DNA-based pairing, A goes with T, C goes with G. So if you match it up, right, AGC is going to go with TGC, and you can check the rest, and the answer is this one. Okay, looks like the ABCD is wrong, but you get the idea. Okay, so these are two rings, and this is one ring. All right. Let me see. There is another one. Okay. This one deals with what happens if you heat up the protein and what structure would not be affected. Okay. So someone asked something like that earlier as well. So I'll let you try that. Okay, I'm going to end the poll just because we're a little bit tight on time. The correct answer is primary. Okay, remember the primary structure is the order of amino acids, right? And they're connected together through covalent bonds. Okay, you cannot break covalent bonds with heat, right? So the answer is A. The secondary, tertiary, and quaternary structures, they are held together by we call them hydrogen bonds, right? So those are not as strong as covalent bonds, and when you heat them up, they are going to break down, and that's what's going to result in denaturation of the protein. Okay. Okay, I'm just gonna tell you the answer for this one. We again running tight on time. In general, what makes one kind of amino acid different from other amino acid? So we talk about the general structure of the amino acid. There's the amino group, there is the carboxyl group on one side, and then there is the R group. Okay, so the R group is what distinguishes one amino acid from another. There are 20 different types of R groups, and that gives the amino acid its property. Okay, so that's it for lecture 2. And I can't believe that took almost an hour. So maybe I skip 3 and 4 for now, and go to number 5. I think number 5 is the one that a lot of you are feeling a little bit antsy about, just because we haven't had a quiz on it yet. So I'm going to do metabolism first, and then when we have time, we will do either 2 or 3. Okay, so metabolism. Okay, so for metabolism, you have to know where things happen. Okay, so there are 4 steps, right? There is glycolysis. Let me just write here, glycolysis. Okay, so what's happening in glycolysis? Let me think about how to do this properly. Let's put box around glycolysis. So we started with glucose, right? Glucose, and then it will go into glycolysis, right? And at the end of glycolysis, it's going to split out. It's going to spit out 2 pyruvate. Okay, 2 pyruvate. Okay, so 1 glucose becomes 2 pyruvate. And what's going to be happening is that we are going to be generating, 2 net ATP. Okay, 2 ATP. Okay, and this is going to be through the process of substrate level phosphorylation. Okay, phosphorylation. So to phosphorylate is to transfer a phosphate onto something. Okay, so in this case, there's going to be an enzyme that takes a phosphate group from a donor molecule and transfer it onto ADP, generating 2 ATP. Okay, and these are net ATP, right? Because you have to invest 2 ATP first in glycolysis, afterwards, you're going to be creating 4 ATPs. So 4 minus 2 gives you 2 net ATP. Okay. So as glucose is being broken down into pyruvate, you are basically ripping electrons away from glucose. Okay. So when you take electrons away from a molecule, it is said to be oxidized. Okay. So glucose is being oxidized into pyruvate. Okay, and in the process, those electrons are going to be captured by NADH. Okay, NAD plus rather, and that is our energy molecule, right? Okay, so I'm gonna put an this really big arrow. I'm gonna put another arrow here. Maybe I'll use a different color for energy carrier. So this one is gonna create the NADH, and there will be a total of 2 NADH. Now, these NADH are going to be created in the cytoplasm. Okay, so this happens, glycolysis happens in the cytoplasm. Okay, and then the electron transport chain is going to be in the cristae, right? In the foldings of the mitochondria. And so what happens is these 2 NADH is actually going to transfer their energy to a FADH2 molecule. Okay, so this gets transferred to FADH2 molecules, and then what's going to happen is we're going to times 2 ATP, which creates 4 ATP. Okay, 4 ATP. Does that make any sense so far?
So if the question is like how many net ATP, we only answer like 2, right? Not the
Yeah, net ATP from glycolysis is 2 ATP, because this is not produced in this is not produced in glycolysis, right? The ATP here, these are produced at the end, right? Okay.
I'm so sorry. And another question. And why is actually then NADH goes to FADH can't that? Can you explain that for me again, please?
Okay, so these two NADH is in the cytoplasm, right? Okay, the ETC happens in the cristae. Okay.
Yeah.
So what happens is the NADH in the cytoplasm does not have access to the cristae, because they would have to cross the outer mitochondrial membrane and then the intermembrane space and then into the inner membrane before they reach the matrix, right? Which is where they have to deposit the electrons onto the ETC. So NADH it's a big molecule. It's not able to cross easily. So what happens is they will, instead of the whole molecule going through, they are just transferring the electron through. Okay, so the NADH will pass the high energy electron to FAD plus, and then that will convert it into FADH2. Whenever you do energy transfer, you will lose some energy in the process. It's never going to be 100% transfer of energy, which is why you cannot transfer electrons from NADH to another NADH. Okay, because you need to lose some energy in the process, right? That's why it's transferring to something that has a lower chemical potential energy. Does that answer your question?
Yes, thank you.
Okay. Everyone else good? Anybody else have any concern questions? We still all here? Yeah. Just checking. Okay, so that's glycolysis. And then what's going to happen is these two pyruvate, if we have enough oxygen, and that's an important part to remember. Okay, if we have enough oxygen, then this is going to proceed to the next step. Okay, and the next step is going to be pyruvate oxidation. Pyruvate oxidation. Okay, so in pyruvate oxidation, you basically making the molecule unstable, so it's going to react, and out of pyruvate oxidation, you are going to get 2-acetyl-CoA. Okay, acetyl-CoA, right? So what are you going to get from here? Well, you are going to get no ATP in the process, no ATP at all, no substrate level phosphorylation, but you will get 2 NADH. Yeah, you will get 2 NADH. Okay, this is per glucose, right? Per glucose, and if you we are already in the matrix in this case, already in the matrix. So we will times 3, right? Because each NADH has enough energy to make 3 ATP. So that will be 3 times 2, which is 6 ATP. Okay. And in the process, you will also spit out some carbon dioxide. Okay, carbon dioxide. Okay, 2 CO2 here. So far, so good. Then what's going to happen is the acetyl-CoA will continue to go forward, and this time you're going to go into the Krebs cycle. Okay, so Krebs cycle is a cycle, right? So it will join with how should I draw this? It will join with something called oxaloacetate. Oxaloacetate. Okay, and then it will combine together with the acetyl-CoA to create citric acid. Okay, you should know this. Okay, if I ask you what is the first product in the Krebs cycle, it is citric acid. Okay, or citrate. In some cases, both are okay. Right, citrate. Right, and then the citrate will go through a series of steps where it will be broken down and it will get regenerated into oxaloacetate, so the cycle can keep on spinning. Okay, so this is happening in the matrix. Okay, so what are you going to get from it? Well, you are going to get quite a bit. you will get 2 NADH. You will get 6 NADH rather. I meant to say for every 2 acetyl-CoA, for every glucose, you will get 6 NADH. You will get, what color did I use earlier for FAD? I don't even know if these colors are helping. We are gonna get 2 FADH2, okay, and you will get 2 ATP. Okay, you'll get 2 ATP like this. Okay, and this is through substrate level phosphorylation. You get 2 ATP. Over here, 6 times 3 is 18 ATP. Over here, 2 times 2 is 4 ATP. Okay, so times 3 times 2. That's how you come up with that. And lastly, you will also be releasing 4 CO2. So together 4 plus 2 gives you the 6 CO2, right? And we have 6 carbon in the glucose, and that's how it becomes carbon dioxide, basically. Okay. And so if you add up all these numbers, right? 2 plus 4 plus 6, if you add them all up, the total is going to be 36 ATP. Okay. So
I have a question.
Yeah, sure.
So for this, like for the Krebs cycle, like not like the glycolysis, we can say, like, if you say like what's the total amount of ATP at the end of Krebs cycle, we could say the
4.
218 and 4, right?
Yeah, 4.
Only 4.
Only 4, because at the end of Krebs cycle, you haven't made any of the the ETC stuff yet, right? Okay.
Okay.
Yeah. So if the question asks from Krebs cycle, how many ATP can be produced in total, then you would count the NADH and FADH2 as well. Okay.
Oh, like if you say total amount. Okay.
Yeah. So I forgot what I was talking about earlier. Right. So that's okay. Other than the 2 ATP here and the 2 ATP here, they are produced by substrate level phosphorylation. The rest of them are produced by oxidative phosphorylation, right? Okay. So I'm not sure how I can do this. Maybe I highlight all these ones. Okay. So these yellow ATPs, they are produced by oxidative phosphorylation. Okay, that's harnessing the oxidative reaction, right? The stripping electrons from glucose, harnessing those energy to power phosphorylation. Okay, so that's like a summary of where everything are. We did some question last tutorial. Where the question is like, if the Krebs cycle spin 3 times, what's the maximum number of ATPs can be produced from that? And we have to calculate it using the NADH and FADH2. So please go through those again. You can watch those, the tutorial recording from last week to see how to do those questions, okay? So that is that. Anything else you guys want me to go through for the metabolism? There is also the fermentation as well as the ETC. But if you guys are okay with that, then I would like to review some of the earlier units instead. Any preference here, guys?
If we can like go through like different kind of diffusion and stuff.
Which one? Sorry.
The diffusion, like the membrane transport, that chapter.
Yeah, I can do that. But any question for metabolism, guys, at all? Let me know in the chat, okay? Like say no question or yeah, move on or not move on. Anything else for here? No. Okay. All right. All right then. Let's move on to the other stuff then. What are we going to do? Let me see. The transport stuff, right? Okay. So that is lecture 4. Lecture 4. There is the membrane protein. Membrane protein. Maybe I'll just quickly review that. So, you know, you're gonna have your phospholipid bilayer here. Okay. Just gonna draw some phospholipids here. And you know, you might have a membrane protein that's gonna be all the way through. And we call those integral proteins. And some of these proteins are very close to each other, perhaps. Okay, so if I have a substrate here, it's going to go into the enzyme number 1, and then it's going to spit out a product, and then that's going to go into enzyme number 2, and then it's going to create a final product. Okay, so this might be enzyme 1, and then enzyme 2. Okay, so that's one membrane protein function, enzymatic activity. You might have a protein here that is going to be receptor protein. Okay, so like if I have a substrate, this is substrate. Okay, and it binds to the receptor here, receptor. Then it's going to lead to change in cellular activity. Okay, and then there are other functions as well. There could be a channel protein, right? Channel protein. Okay, so this is going to allow maybe charged particles like calcium or something to go through. Okay, so this is channel protein for transport, right? Transport. We can also have let me think as I draw this stuff. We can also have a protein that's just kind of on one side, right? Not span the entire length of the phospholipid bilayer. That would be a peripheral protein, and it's going to be attachment to cytoskeleton. Okay, so if it's on the inside, then it helps maintain cell shape. If it's on the outside, then it helps the cell move around in the extracellular space. Okay. Again, more phospholipid bilayer there. We can have one with sugar on it. We talked about this earlier, right? These are sugar, right? And this is for cell recognition, right? So cell recognition. Okay, and this is a glycoprotein. And finally, we also have this would be like, you know, cell number 1, and then you might have another cell here. Okay, you might have another cell here. Sorry, another cell here or something. Okay, and then they would also have a similar protein. Okay, another cell here. Okay, so this is cell number 1, cell number 2, cell number 1, and this is called a cell junction. Okay, so that basically summarizes all the cell function. Yep, the cell ID would be the cell recognition. That's correct. Okay, Edward.
So but like if it says tight junction, is the same thing?
Yeah, tight. It's a tight junction is a type of cell junctions. Okay, there are different types of cell junctions. Yeah, and you know, we might have some you know, cholesterol here, right? Cholesterol here and there. Okay, so these are cholesterol, which would modulate the fluidity of the membrane, right? So the more cholesterol you have, the more fluid it's going to be, the more leaky the cell is going to be. So one of the questions that I was asking in the lab was that name two factors that, two reasons why high temperature is going to result in more pigment leaking out. And the two key things that you needed to tell me is that number one, the heat is going to cause more movement in the phospholipid bilayer, okay, because they have more kinetic energy, right? And that's going to make it more porous. And the second key thing is, as temperature continue to increase, when we get to the 80 degree, right, or 60 degree and beyond, that's high enough temperature for the membrane proteins to start to denature as well. So once the proteins denature, that really disrupt the integrity. So that's why there is a sudden sharp increase in intensity of color beyond 60. Okay, so if you just disrupt the phospholipid bilayer a little bit, you know, between 40 and 60 range, you will get some color out. But once the membrane proteins are degraded, then it becomes super leaky, and there's going to be a lot of pigments coming out. Okay. So that is the membrane protein. Okay. I'm just going to take quick 5 minutes. I need to get something to drink, and then we will resume afterwards. Again, please type questions in the chat box. I don't know what you don't know. Okay, so I will look at that when I come back. Okay, so 5 minutes. We'll resume in 2:23. Okay, let's keep going. Discuss about some application questions. Nazneen requests that. Yeah, we will do some more polls as we go on. Okay. Am I recording? Yes, I am. Okay, so the next thing we're gonna do is the membrane protein. For the membrane protein lecture is the type of transport, basically. Okay, so let's try to summarize this for you. Okay, so we have transport. Okay, so transport is separated into active transport, and also passive transport. So the active transport is going to require energy. Okay, usually in the form of ATP, but not always. Okay, so require energy of some sort, and it's going to be against concentration gradient. Okay, against concentration gradient. Whereas for passive transport, it is going to require no energy required, and it's going to be down the concentration gradient. Okay, so those are the big difference between active and passive transport. So under passive transport, we have various types. Okay, under passive transport, for example, there is osmosis. Okay, osmosis. Osmosis is diffusion of water, and it's for water only. Okay, so you should review. I mean, I can do that too if we have time. We should review hypo, and isotonic. Okay, that has to do with osmosis basically. Another type of passive transport is facilitated diffusion. Okay, so in facilitated diffusion, the molecule is either too big or it cannot interact well with the phospholipid bilayer. So you require the assistance of a membrane protein. And there are two types of facilitated diffusion. which include carrier protein and channel protein. Okay, so carrier protein is typically for larger molecules such as glucose. Okay, so they cannot squeeze themselves through the gaps in the phospholipids. They're too big, and so you need a carrier protein to move them across. Whereas channel proteins are usually ions. Okay, usually to transport ions. So example would be something like, you know, Na+ or something. Okay, they cannot get through because they're charged, and so you would require a channel protein. And the last type of passive transport would be simple diffusion. Simple diffusion. Okay, so these are best for small non-polar molecules. Okay. So those are the three types of passive transport. Over here, we have the active transport. Okay, active transport. So active transport, we have protein pumps, and bulk transport. So protein pumps, we talked about the sodium potassium pump in lecture, but later on, when we, like lecture 5, we learned about the ETC, the electron transport chain. And that actually is an example of active transport. The only difference is we're not using ATP as the energy source. We're using the high energy electrons from the energy carrier to pump the protons from the intermembrane space, from the matrix to the intermembrane space. Bulk transport, there is endocytosis. There is exocytosis. Okay, so endocytosis is to move in a large amount of material into the cell, whereas exocytosis is to move a large amount of material out of the cell. And that is going to have three types of endocytosis. There's phagocytosis, which is taking in some kind of solid particle, like eating a bacteria, for example. There is pinocytosis, which is taking in liquid primarily. I mean, there is going to be some dissolved ions and stuff like that, but the main goal is to take in liquid. That happens in, in the kidney cells. That's also how infants are able to absorb the milk in their digestive tract. And the endocytosis also have a third type, which is the highly specific one. Okay, this one is called the receptor mediated endocytosis. Okay, so that is those are the different types of transport. Okay. In terms of the three types of tonicity, right? So here we have the cells, okay, in a beaker. Okay. Let me try to do this. So if the concentration on the inside is same as outside, then it's isotonic. If there's more stuff inside than outside, then it's hypotonic. And if it's the other way around, then it's hypertonic. Okay, so let me just write this down for you. This is isotonic. It is best for animal cells. Okay, this one is hypotonic, okay, because the water is gonna come in, right? There's more water coming in than water leaving, and the cell is going to swell and burst, right? So lysis could happen, and unless it's a plant cell, okay? So, oops, what happened there? So this is best for plant cells, okay? Because they have the cell wall, okay? They're not gonna lyse. So lysis for animal cells. Animal cells. And this one, you will have more water leaving than coming in. And it's going to cause the cell to shrink, and we call this hypertonic. Okay, so plasmolysis. Okay, or another word for that is shrinking. Okay, that is basically the transport unit. Let's do a few more polling questions. There we go. Okay, please try that. Okay. The correct answer is facilitated diffusion. So these are like macromolecules, right? We're talking about proteins, right? Endocytosis would be for something like an entire cell, right? Like if you're trying to eat like a bacteria, like I said, right? Or if you're trying to bring in large amount of fluid. So for proteins that are too big, definitely facilitated diffusion. Okay, this is for the metabolism unit. So let's try this. Okay, I'm not sure where everyone is. Only 11 people responded, but that's okay. The correct answer is all of the above. Okay. I think there is a slide in the lecture notes. It's called the versatility of the cellular respiration or something. I forgot the exact title. But that slide shows that all these things can actually feed into various points of the cellular respiration. Okay, so glucose is the preferred source for production of ATP, but all the other things can be used as energy sources as well. Okay, let's do another one. This one should be straightforward. Okay, almost all of you got it. It is the NAD+, which will pick up the electron and become NADH. Okay, let's do one of a harder question here. Try this, please. Okay. The answer is A. Okay, so this you just have to look up the alcoholic fermentation pathway, and you will see that acid aldehyde is going to be reduced into ethanol. And let's say you don't remember, right? Let's say you don't remember the pathway on a test situation. You can still think about it from a different approach. So here, NAD plus is regenerated from NADH. So going from NAD plus to NADH, you are picking up electron, which means the other way around, NADH to NAD plus, you will be losing electron. Okay, so if let's see if I can write here, if NADH becoming NAD plus involves losing electron, right? Losing electron, then this is going to be oxidation, right? Then that means whatever else is happening has to be a reduction reaction. So it's either A or C, okay? And this is talking about alcoholic fermentation and not lactic acid fermentation. So that's why the answer is going to be, it's going to be A, okay? So we have a little bit more time left. Maybe we will do just a quick summary of lecture 3. I think lecture 3 is probably one of the most more straightforward lesson. It's just about organelles and stuff like that. But since we have time, we can quickly review that as well. So maybe we do another chart. So we can do types maybe, and then we have the organelle, and then we will just do main function here. Okay, main function. So membranes, organelles rather, it's separated into two categories. You could say that. Okay, we have organelles with membrane. Okay, and then organelles without membrane. So let's think about some organelles that has membrane. Okay, the nucleus, right? The nucleus has membrane, and the function of nucleus would be, protect and we can say control access to DNA. Okay, so I am purposely using slightly different wordings than what we already did in the study guide. So you will have a different perspective, I guess. Okay, and another key function of nucleus is because it contains the nucleolus. That's where you are going to be able to synthesize ribosome. Okay, synthesize ribosome. So the nucleus is membrane-bound organelle. And then the next one, we have a rough ER and smooth ER. Okay, the rough ER is a direct continuation from the nuclear envelope, and then the smooth ER is a direct continuation extension of the rough ER. So rough ER is for protein synthesis. Okay, and again, right, you want to think about where these proteins are going to end up, right? These proteins can end up in the membrane. The proteins can end up in the cytoplasm. It could be in a vesicle waiting to be released, right? It could become a lysosome, or it could be something that is going to be released immediately. Okay, whereas the smooth ER is primarily for lipid production. Okay, lipid synthesis. Okay, so there was a lot of questions on quiz 2, right? About if a cell has a lot of whatever, what is the likely function, right? Okay, so that deals with a lot of these. It's basically asking you the function of the organelles. Okay, so if you have a lot of smooth ER, right, then the cell is likely to be making a lot of lipids. Right? We can rephrase it in the other way, right? I think the question was like, it's a cell that is making a lot of steroid hormones, so you would expect it to have a lot of what? And steroid hormones is a lipid, so you would expect it to have a lot of smooth ER. Okay, so you might see some of those questions again, right, in the test. In fact, we took up some questions, right, at the beginning of each of the tutorial. So those questions are worth revisiting again in preparation for the test. Next, we have the Golgi body. Right, Golgi body. That is for modified contents of vesicles, right? New proteins, right? Sort and ship proteins. And we have lysosome. Okay, lysosome. Lysosome is in one simple word sentence, basically is carries out cellular digestion. Okay, so you either digest all and worn out organelles, or you digest things that you are ingesting. Okay, and so these things, they are part of the endomembrane system, right? I'm going to put star here. Part of the endomembrane system. Okay. So other things that have membranes, but not part of the membrane, the endomembrane system, include things like mitochondria. Okay, so that is energy production. You should know that by now. Definitely know the equation for cellular respiration. Know what's reduced and what's oxidized. In plants, we have chloroplast. Okay, and that is the site of photosynthesis, right? Site of photosynthesis. And we have peroxisome, which is used for inactivates toxin. Okay. So, and there's one more that I can think of, vacuoles. Okay, so we don't have vacuoles, right? It's only in plants, right? So this is for storage of water, nutrients, right? And you know, all the good stuff we talked about in lecture, pigments, maybe toxins and whatnot. Okay, so these are all organelles with membranes, and then we have organelles without membrane. Without a membrane. Okay. Anybody know what they might be? I can only think of, like, you know, a few.
Like ribosomes and
Yeah. Ribosome is a good one. So that's for protein synthesis.
Cytoplasm.
Cytoplasm is a membrane itself, and you know, typically that's not considered as an organelle. Anything else like inside? Yeah, okay. Let me finish this first. Synthesis. Okay, so in the chat, someone says cilia and flagellum. Yeah, cilia, flagellum, flagellum. That's for movement. I actually did not think of that one. I had something else in mind. So thank you for mentioning that. Cytoskeleton is it's not really organelle. It's kind of like just, you know, protein in the background, right? Per se. So not really an organelle.
so I have a question.
Yeah.
So it's like lysosomes like come from the Golgi body, right? So can
Lysosome comes from the yeah. So that's part of the endomembrane system. Yeah. Okay. So Samin says centrioles. Yeah, that's the one I was looking for. Okay, centrioles. That is to make microtubule, right? Microtubules. I think in the lecture I call them spindle fibers. There are different types of microtubules. Okay, the ones that are created by centrioles, they are called spindle fibers. Make microtubules to guide chromosome movements during cell division. Okay. I think that pretty much you know, summarizes everything that we've done from 2 to 5. I mean, sure, I did not cover, you know, every possible topics, but these are the key ones. And you know, it's good to go through those study guides again that we did every week in the tutorial. It's not difficult, the test, okay? The test is, like I said, lecture 2, 3, 4, you've already done the quiz. So the questions are taken from the same pool of questions as the quiz. So you might even see some questions that are, you know, repeating themselves. So it's like, I'm not trying to, you know, make it super difficult for you guys. And the class average has been actually quite well. So, you know, I'm not particularly concerned, and you shouldn't be, as long as you study for it. So that's it for the first half of the course. Next week we will do the second half, which is lecture 6. So next tutorial will be again on Thursday, and you know, it's going to be more problem solving from now on for the second half of the course. There is still some memorization, but you know, there is more application, I would say. Not application in terms of like application questions on tests, but like kind of like a math problem and chemistry problem where you have to solve something. Okay, we'll be, you know, looking at genetics. We'll be looking at transcription, translation, you know, biotechnology, that kind of stuff, which is all going to require a little bit of applying what you've learned. So there's a shift in paradigm in that respect. Okay, so if you don't have any other questions, good luck. Study hard, and I'll see you next week. Like I said, I will post this lecture recording, and I will post the note that I have here as well. Okay. Have a good one, guys. Bye now.